Hello students, today's topic is about an important signaling molecule, that is nitric oxide, and this is the third lecture within this signaling classes. And before that we have just gone through the GPCR pathway, trimeric GPC proteins, and the last lecture was around IP3 DAG pathway. So today we have come up with yet another pathway, that is NO, that is nitric oxide as signaling molecule. Now this nitric oxide does have very important clinical implications. I would try to go through that in the last phase of the lecture, but now let's start with the basics of the signaling pathway. So this starts when a ligand like acetylcholine, binds with the receptor which you see right on your screen, is that is this GPCR, G protein coupled receptor. And once this ligand binds with this G protein coupled receptor, it activates an important trimeric protein, that is G protein, and out of that it is the G alpha subunit of that protein which gets activated. And you know the basic fundamental of that pathway is that when G alpha subunit is bound with the GDP, it is in active form, but whenever the signal is transmitted to the G alpha subunit, this G alpha subunit does modify itself by replacing the GDP with what? GTP. And whenever there is the GTP bound form of G alpha subunit, it is always going to be active. So this is what we have shown you right here in this example. So what is this G alpha subunit bound with the GTP? It is in the active state, and this alpha subunit then in turn goes and activates what you call as the phospholipase C. So PLC is what? It is an important enzyme, that is phospholipase or lipase C. So this is PLC. Let me recapitulate for you. There is receptor plus ligand. So ligand that is bound to the receptor, that leads to the G protein coupled receptor activation, which in turn activates G alpha subunit because of the conversion of GDP to GTP, and this ultimately activates protein, that is phospholipase C. So what this phospholipase C does is it cleaves or breaks this important PIP2, that is phosphatidyl inositol 4,5 bisphosphate. So this PIP2, it is being cleaved into two important products. What is that? The first, that is diacylglycerol, and second important product is IP3. So this is the basics till now. So receptor plus ligand, GPCR activation, phospholipase activation, and ultimately PIP2 getting cleaved into DAG and IP3. Now let me first erase all of these things to make the screen doesn't look messy. Now what this IP3 does is IP3 goes to the endoplasmic reticulum and opens up this channel, which is IP3 gated calcium channel. So whatever the calcium that is stored within the endoplasmic reticulum, that comes out into the cytoplasm. So this is the calcium molecules that are present within what? Cytoplasm. So within what? The cytoplasm, there is another important molecule, that is calmodulin. So this calcium and calmodulin together interact and form what you call as calcium calmodulin complex. So very simple. So ligand plus receptor, G protein coupled receptor activation, G alpha subunit activation, PLC ultimately breaking down PIP2 into IP3 and what you call as DAG, IP3 then activating calcium, release of the calcium from the ER, and together they form calcium calmodulin complex. And ultimately this calcium calmodulin complex does have a very fundamental role, because this activates an enzyme, that is NO synthase. What is NO synthase or nitric oxide synthase does? It converts arginine, that is arginine plus oxygen, into citrulline or citrulline, whatever you pronounce, plus nitric oxide. So this is the actual molecule that we are talking about. So this nitric oxide is only and only synthesized when there is this synthesis of NO synthase. But this NO synthase won't be formed unless and until there is this calcium calmodulin complex. And for the formation of the calcium calmodulin complex, you need the release of the calcium from ER. That is only possible when there is IP3, and IP3 forms when PIP2 is cleaved by PLC. And PLC comes because of the G-alpha GTP subunit, and this is all because of the ligand and receptor interaction. So if you are clear about that, then we are going to go with the role that NO plays in the cellular functioning. This NO then diffuses out of this cell, this endothelial. This is primarily your endothelial cell or blood vessel, endothelial cell. And surrounding the blood vessel, endothelial cells are what you call as the smooth muscles. So these smooth muscles do surround the endothelial cells, and this NO molecule then comes out and binds with a receptor that is what? That is NO receptor. And once it binds, this ultimately leads to the conversion of GTP into cGMP. So within the smooth muscles, this NO molecule helps in the activation of cGMP, and cGMP then activates the protein kinase G, and this protein kinase G ultimately helps these muscles relax and help in what you call as vasodilation. So this is the basics of the NO signaling. So if you just want to go through this, you have everything right on the screen. You can take the screenshot or just pause your video, replay, rewind, whatever you want. And this pathway plays a very fundamental role in the patients who have what you call as the angina. So angina patients do, doctors do recommend what you call as the nitroglycerin. So what this nitroglycerin does is it actually helps in the synthesis of NO. NO then goes to the blood vessels, and help them what you call as relax or dilate. And when there is vasodilation, there is increased blood flow to the heart, and that patient then feels better. Angina is because there is the decreased flow of the blood flow to the heart. And in case of the men, the erection of the penis is only possible because of the synthesis of nitric oxide. And this nitric oxide, because of the same mechanism that I have shown you right there a couple of minutes ago, that NO vasodilates the arteries that are supplying the blood to the penis, and there is the greater or increased blood flow to that particular organ, and that helps in what you call as the erection. So there is one point that I want to make, that is